Thank you for inviting me. Today I will discuss the advantages and challenges of broad molecular profiling. This is my disclosure. As you can see, I have received speaker honoraria and also non-financial support from Thermo Fisher Scientific, which is the provider of the oncoming comprehensive plus assay, which I will discuss during my talk. So I would like to start the discussion about genomic profiling in general and why it is so relevant nowadays. As we know from clinical practice, and as you see here from this slide, also from clinical trials and their current design, we went from enrolling patients exclusively based on the tumor type to having a nice selection of biomarker-defined patient populations, either within specific tumor types, in umbrella trials, as you can see here in the middle section of this figure. For example, for HER2 amplifications as a biomarker, we have trastuzumab as a drug. For ALK translocations, specifically in lung cancer, we have crizotinib as a drug, and for BRAF mutations in melanoma, we have vemurafenib as a drug. But we also, very importantly, can select just based on molecular abnormality, and we can have this agnostic, histology agnostic approval of drugs nowadays, which happened, as in the last example, for microsatellite unstable cancers, so in general solid tumors that are mismatch deficient, mismatch repair deficient, and that can receive pembrolizumab, so immune checkpoint inhibitors. However, here I just spoke about 4 of such molecular biomarkers. Why do we need comprehensive genomic profiling? The number of these biomarkers is increasing every year. Here you can see a picture showing just a few representative biomarkers for the situation that evolved in the last 20 years, and you can see that, for example, in the last 6 years, we have more than one biomarker-based therapy that was approved or that became relevant in the clinical field. And of course, this is great news for patient management. However, it is also required that clinicians perform broad molecular testing to be able to assess all these different biomarkers. And now I would like to again stress this point. So the so-called actionable genes, so genes that are directly associated with treatment and for which there is a drug that is FDA approved, are not so many. During my talk, I will talk about next generation sequencing assays. And here you can see there are only 55 genes that are therapy approved for all cancer types. And 55 target genes can be easily covered by small next generation sequencing assays. So why do we need comprehensive genomic profiling, so broader next generation sequencing? Here you can see that the picture is different if we look at genes that are instead associated with a clinical trial. There is this magic number, which is close to 500 genes that are associated with novel therapy options. And of course, you can imagine that this information is very valuable when there was no finding of molecular alteration in the previously mentioned 55 or so genes and in advanced cancer settings. Moving to clinical research, what are the options for comprehensive genomic profiling? So the furthest we can go is whole genome sequencing, which, however, is time-consuming from the analysis point of view and also very expensive. However, it allows to sequence the whole tumor genome. If we want to go a little bit less broad, we can sequence only the coding part of the tumor genome with the so-called whole exome sequencing, which only sequences exons. And this is, of course, a bit less expensive and time-consuming, but still on the broader or larger side. And then we have clinical exome sequencing, which instead is consistent sequencing only clinically relevant or, let's say, cancer-related exons. However, here we are still talking about expensive methods that also required really time-consuming analysis. What has come up in the last 2-3 years is actually targeted sequencing approaches, which anyway are broad enough to allow comprehensive genomic profiling. And here you can see 2 examples on the right side of my slide. We have the TrueSight 500 oncology assay from Illumina, and we have the Oncoming Comprehensive Assay Plus from Thermo Fisher Scientific. I will now share with you our results obtained using the Oncoming Comprehensive Assay Plus, and here is an overview of this assay. It includes 500 genes that can be assayed from both DNA and RNA levels. So we can inspect single nucleotide variants, index, as well as copy number alterations from the DNA point of view, and then we can have analysis of fusion and splice variants from the RNA side. This assay also allows to assess immuno-oncology research targets, as are the famous tumor mutational burden or microsatellite instability. And very importantly, because it is for oncology research, it is compatible with FFP tissue, and it only requires 20 nanograms of input material. And we know that nowadays cancer specimens, biopsies become smaller and smaller, so this is a very important point. It is also based on the traditional oncomine workflow. Thus, if this workflow is already available in the laboratory, it is easy to implement it. I also would like to stress that among these immuno-oncology research targets, there are also a nice collection of genes that have actually been associated in clinical research with response or resistance to, for example, immune checkpoint inhibitors. Here you see at the bottom of this list STIC11 and KEAP1, which together with KRAS alterations have been associated with resistance to immune checkpoint inhibitors. And on the top of the list, you also see several genes that are associated with antigen presentation, and also here, again, mutations, loss of function mutations, for example, in beta-2 microglobulin have been associated with resistance to immunotherapy. Here, I want to show you our results. We have used the Oncoming Comprehensive Assay Plus to profile 25 cancer samples from which we had that were previously characterized in our lab. And here you can see the results obtained on 6 of these samples, 4 non-small cell lung cancers and 2 colorectal cancer samples that were profiled with one of the so-called small, or yes, I would say the smaller targeted NGS panels, the oncomyte solid tumor DNA, which is also IVD labeled. And you can see here that we found 13 mutations across the 6 samples, and some of which very clinically relevant. As you can see at the bottom of my slide, we found 3 mutations for only 1 sample in EGFR, exon 19 deletion, T790M, and 797. And these mutations really are crucial for or are associated with resistance and response from IGFR tyrosine kinase inhibitors. And you can see that the oncoming comprehensive assay plus performed well as our smaller panel. So there is 100% sensitivity and positive predictive value. So all mutations were detected by the assay. If we go a little bit broader instead, so we have analyzed 17 non-small cell lung cancer samples with the oncoming tumor mutational load, which is actually used for tumor mutational burden estimation, and we have validated it in our unit for this purpose, as well as for variant calling if samples are sequenced at an appropriate depth. And here you can see we have found 121 mutations across the 17 samples, and again also here sensitivity of the oncoming comprehensive assay was 99%, and also positive predictive value was very high. To continue talking about the tumor mutational burden, we analyzed the 17 non-small cell lung cancer samples in a retrospective clinical research study, and we had information about a response status from immune checkpoint inhibitors, and also PD-L1 status assessed by immunohistochemistry for these samples. And you can see this information summarized here in the plot that is in the middle of the slide. So this is an oncoprint, and each column represents one sample. On top, you can see the green samples showed durable clinical benefit, which corresponds to progression-free survival longer than 6 months. And the gray samples on the right side of the plot are instead no durable clinical benefit observed. You can also see the oncoming comprehensive assay plus allows to assess the microsatellite instability status. However, these were all lung cancer samples, so they all were microsatellite stable. We had, as I said, PD-L1 status information, and you can see here the TPS score represented by purple gradient color. And you can see tumor mutational burden, TMB, as assessed by the oncoming comprehensive assay plus. So very interestingly, you can see the darkest shade of orange represents TMB high patients, and you can see that 2 patients out of 8, two samples out of 8 durable clinical benefit were actually TMB low. However, all other samples at TMB high. And for the non-responders instead, also here we have 2 samples that were TMB high but showed no response. If we go and look at the mutational status of these patients, we also can see some interesting features. So as I discussed before, STIK11 and KEAP1 mutations, especially in association with KRAS, have been linked to resistance from immune checkpoint inhibitors, and indeed we did find them only in our no durable benefit cohort. Talking about TMB, I also want to share with you a correlation that we have done with the non-orthogonal test for TMB estimation. Here you can see actually TMB as assessed by Foundation Medicine, and this test actually, or this correlation was done with only 6 samples. However, the results are, I would say, pretty good, and this is important because harmonization of TMB estimation between different assays is critical, and it's been very discussed recently. We also characterized using the oncoming comprehensive assay plus 8 colorectal cancer samples that were previously tested with the Bethesda panel for microsatellite instability, and they also were tested via immunohistochemistry for mismatch repair protein loss. And here you can see a representative immunohistochemistry of the 4 proteins that are usually tested, and of which loss indicates mismatch repair deficiency. And you can see here that in the middle plot, again, each column representing one different sample, we had 3 samples in orange that showed loss of expression in one of the mismatch repair proteins. And very nicely, we could recapitulate that loss by finding truncating mutation in the corresponding protein using the oncoming comprehensive assay plus. As I said before, the assay allows to estimate microsatellite instability by giving a microsatellite instability score. And you can see here that the microsatellite instability score correlated quite well with the number of microsatellite markers that were unstable according to the Bethesda panel. We also had TMB estimation. I would say TMB also correlates quite well with the microsatellite score and with the Bethesda panel, number of markers unstable, however, not in all cases. And if we look at the nice collection of mutations that we have detected in these samples, you can also see that, very importantly, there are two samples that show truncating mutation in beta-2 microglobulin, and this mutation would speak for resistance from immune checkpoint inhibitors. However, these samples are also MSI high, actually, samples, which would instead predict response from immune checkpoint inhibitors. Therefore, these findings have to be interpreted with caution. I would also like to give you an outlook on our future plans. We also want to validate another feature of the oncoming comprehensive assay plus. We are particularly interested in this feature, and it is about copy number alterations. So NGS is routinely used for mutations or variant detection in cancer specimens. However, copy number variation through NGS is not yet been established, and the current methods are mostly arrays. So, for example, SNP arrays or DNA methylation arrays most recently. And however, as you can see, the oncoming comprehensive assay plus has an impressive collection of genes, and the amplicon design allows actually to detect for specific CMB gains and losses. Therefore, we want to test this feature, and we plan to include 40 cancer samples with known clinically relevant copy number variations. And as orthogonal methods to validate, we plan to use the Illumina Infinium DNA methylome array as well as the OncoScan CMB Plus kit. I also want to focus on one more feature of the panel, and this is regards the analysis of the data. So analysis of comprehensive genomic profiling data can be very cumbersome, and instead this assay offers an integrated solution through the Ion Reporter software. And I think this is a very big advantage because it is automatable, but is also a customizable option. And you can see here actually that the interface is very intuitive and user-friendly. So we can have immediate information about tumor mutational burden and MSI score, as well as the mutations that are detected. And there is also the possibility of setting customized filter chains to just see mutations that pass specific QC parameters. So to conclude, I would like to briefly discuss the advantages and the challenges of comprehensive genomic profiling. Between the advantages, we can have a higher probability of identifying relevant biomarkers by doing broader genomic analysis. However, there is also a higher probability of detecting variants of unknown significance as well as therapy non-relevant information. The bioinformatic workflow is evolving to simplify the analysis. However, as I just said, the analysis of broad genomic profiling results can be cumbersome and much longer than for targeted and smaller next-generation sequencing assays. It is a one-stop shop option, so we can see genomic alterations as well as TMB and NFI status. However, this might not be required in all situations, so it is important still to pay attention to the clinical context. And in conclusion, I also would like to say that this assay is so broad that it can be used independently from the cancer type, so it fits different cancer types, so it can be used as a standardized laboratory procedure. However, of course, it requires higher number of sequencing reads, therefore higher price, and most of these assays also require higher input DNA. However, this is luckily not the case for the oncoming comprehensive assay plus. So finally, I would say that comprehensive genomic profiling in general will prove very important in the context of immunotherapy. You can see here that there are really several biomarkers that are being studied, and some of them are already being used in the clinics to address the possibility, the probability of response from immunotherapy. And I believe that comprehensive genomic profiling will support this by allowing to account for several biomarkers and their interaction, and potentially allowing to better precision in patient stratification. With this, I would like to thank you for your attention, and I also would like to thank the Molecular Assay Development Unit of the Institute of Pathology of Basel, as well as our clinicians that supported these studies, and Thermo Fisher Scientific. Thank you very much.